Okay, good afternoon. So today we will continue dealing with the nervous system block. We studied senses, general senses, and started studying the special senses. Last time we dealt with three of them: taste, olfaction, and vision. And today we are going to continue with the last two, which are hearing and equilibrium. And you will see that they are so closely related to each other. Their receptors are located almost in the same, they're actually present in the same bone, and they are served by two divisions of the same cranial nerve, the vestibulocochlear nerve. So to start with, I will deal with the hearing, and we will start with the anatomy of the ear. And the ear is actually divided into three parts. It's not only this part that you can see on your side of your head. This is only the external ear, but we have also the middle ear, the blue one, and the inner ear, which is located here, the pink structure, and they are all located within the temporal bone. So the external ear, generally speaking, consists of the auricle and the external auditory meatus. It collects the sound, and then the middle ear, it amplifies this sound mechanically, and then the inner ear, the general function of the inner ear is that it converts these mechanical waves, it converts them into electrical waves, into nerve impulses, and sends them to the brain. So it has the specific sensory receptors, and it's a very complicated structure. That's why they are considered the special senses. Let's start with the external ear. What we see here is called the auricle, or the pinna of the ear, and probably we have mentioned that previously, that it consists mainly of cartilage, which is an elastic cartilage. That's why it can recoil back to its shape when we bend it. And then it's covered by skin, and it leads into a canal called meatus, or a canal, external auditory meatus, or external auditory canal. And this consists of two parts. The outer part, as you can see here in blue, which means cartilage. So the outer part is in cartilage, and then the inner part of the canal is bony. It is within the temporal bone. It is lined by skin, this external ear, and contains hairs and contains specialized sweaty glands called ceruminous glands. So they are specialized sweaty glands. They produce the earwax. Of course, the presence of the wax, the presence of the hairs will prevent the passage of foreign substances into the ear, although sometimes they might pass and need to be sometimes removed by the physician or by the surgeon. And also the presence of the earwax is important in that it is a water repellent, and not only a water repellent, but it is a repellent of insects. So it repels the insects and prevents them from entering and housing the external auditory meatus. Some people might say that if you are annoyed by a mosquito, then take part of your earwax and spread it on your skin, and it is a mosquito repellent. And it leads at the end to the tympanic membrane, and this is very thin, semi-transparent membrane. If you look at it through a scope, which is called otoscope, like we mentioned previously, there is an ophthalmoscope. We can look through the pupil to see what is the inside of the ear. Again, you can look and see what is inside, and using an otoscope. It is thin, semi-transparent, and it is not flat, as you can see. It is concave, and it is covered on the outer side. It's covered by epidermis, and then there is a sandwich of connective tissue deep to it. One point to be mentioned here is that the external auditory canal, or meatus, is not straight. It is a little bit curved, and usually it is directed forwards and medially. So that's why the tips of the stethoscopes, they are not quite horizontal, but they are directed anteriorly, so that to fit, when you put them on, they fit on the external auditory meatus, and they cannot fit if you put them the other way around. So they are not only horizontal, but they are directed forwards and anteriorly to fit into this curvature of the external auditory canal. Then we have the middle ear cavity. You can see it here. It is enlarged, the middle ear cavity. It extends between the external ear, external auditory meatus, and the inner ear. And it is separated, of course, from the external ear by the tympanic membrane or the eardrum, and is separated from the inner ear by two windows. We will deal with them in details in a moment. One of them is called the oval window, and the other is called the round window. And the middle ear cavity contains air. So there's no fluid here, it is air. And as you can see that there is a communication between the middle ear cavity and the nasopharynx, the part of the pharynx that is located behind the nose, the upper part of the pharynx. So there is a communication through a tube, which is called the auditory tube, or it is well known as the eustachian tube as well. It communicates between the middle ear cavity and the nasopharynx and help equalize pressure, air pressure, in between the external environment and the middle ear cavity. Otherwise, as probably you have experienced that before, during takeoff or landing, for example, you will feel that there's something annoying your eardrum because the pressure changes in the cabin are so rapid that the changes in the pressure in the middle ear, so the pressure changes here are very rapid, but they are slow here in the middle ear. It takes time until this equalizes the pressure. And in order to overcome this, probably you have discovered that if you swallow, then this discomfort will be relieved soon, because while you are swallowing, there are small muscles attached to the auditory tube, to the eustachian tube. And by the way, the eustachian tube is like the external auditory meatus. Part of it is bony and part of it is cartilaginous. So this cartilaginous part can open and close, and this usually happens. It opens during swallowing, and this will push more air into the middle ear cavity to equalize the pressure. Some companies usually they distribute like candies before landing or before takeoff, so that while you are putting them in your mouth, you are swallowing and this will help you to avoid the discomfort that might take place because of unequal pressure on either side of the eardrum. Now, inside the middle ear cavity, there are three bones, and the names of these bones in Latin is derived from their shape. So we have the first one is malleus. Probably we mentioned them as part of the axial skeleton in the lecture on the axial skeleton. So the malleus, which means like a hammer, and then we have the incus or the anvil, and then the stapes, which is like a stirrup. It has a foot plate here, oval foot plate. This is the stapes. To remember them from lateral to medial, MIS, miss, malleus, incus, and stapes, and they are connected to each other by synovial joints. The presence of synovial joints indicates that there is good amount of mobility. That's why we have synovial joints. As we mentioned that previously, synovial joints are always present when there is good amount of movement taking place. So you can see that the malleus has a handle which is attached to the tympanic membrane. It's fused to it, attached to it, malleus. And the stapes has the foot plate, which is attached to one of the two windows that separate the middle ear from the inner ear. And because the stapes is oval in shape, then it closes what we call the oval window and leaves the other window here, which is round window. This window is covered by a curtain or a fibrous tissue, let's say. It's called the secondary tympanic membrane. In addition to that, the middle ear also contains muscles. Just to let you know that one of the functions of the middle ear cavity is that to amplify the sounds that are, the vibrations that are present here, and the sound vibrations of the tympanic membrane, they amplify them because of the lever action of the malleus, incus, and stapes on one hand, and because of the fact that these waves are going to be transmitted to waves at the oval window, and the oval window is actually about 20 times smaller than the tympanic membrane. So the sounds are getting amplified by the lever action of the movements of the bones and by this disproportionate size of the tympanic membrane and the oval window. So in other words, we need mechanisms to protect the ear from very high sounds. That's why we have two muscles. These two muscles, they are skeletal muscles, but they act reflexly, okay? They are so tiny, small muscles, you don't expect that you can easily see them. And one of them is called the stapedius muscle, and it's attached to the stapes, okay? And it prevents large vibrations in the stapes from loud noises. And the other one is called the tensor tympani. This one, tensor tympani. It's attached to the malleus, to the handle of the malleus, which is attached to the tympanic membrane. So again, it tenses the tympanic membrane and it prevents excessive vibrations of the tympanic membrane. So you can expect if these muscles or one of them is paralyzed, then the patient will have high sensitivity to loud noises. It's called hyperacusis. The patient will hear like normal sound as shouting. So two muscles are also located here, and three bones and the ear. And there is the auditory tube. Let's deal with the inner ear now, and it consists of two labyrinths, bony labyrinth and a membranous labyrinth, okay? So this is the bony labyrinth, and actually, if you look within the skull, you will not find this structure. This is a cast of canals and caves that are present within the bone. So it's not present as such, but this is the cast of these spaces. So they are spaces within the bone. And the bone is called the petrous part of the temporal bone, the rocky part, which looks like a rock, but inside this rock, inside this mountain, there are caves and tunnels. And this is how they look if we have a cast of them. And so this is what we call the bony labyrinth, and therefore it is lined by periosteum, and it contains fluid, partly it contains fluid which is called perilymph. And this perilymph is almost the same as the cerebrospinal fluid. And the reason is that it's going to protect another labyrinth which is located inside, and this is called the membranous labyrinth. And it is the membranous labyrinth, as you can see it here, within the bony labyrinth, that contains the hair cells, the sensory cells. So they need a lot of protection here. They are present in a membrane, and they are surrounded by fluid, and then the membrane is surrounded by fluid, and then that fluid is surrounded by bone, by the bony labyrinth. And so the bony labyrinth consists of, as you can see, semicircular canals. They look like circles, but they are incomplete circles. So they are semicircular canals. And there's a region here, which is called the vestibule. And then we have the cochlea, which looks like the shell of a snail. So that's why it's called the cochlea. The receptors for hearing are present here, while the receptors in the cochlea, while the receptors for equilibrium are present in the semicircular canals and in the vestibule. In fact, they are not present in the bony part, they are present in the membranous part, which is present within. So the membranous part, you can see it here. I will talk about it here in this slide. So the membranous labyrinth, it is again membranes. They have the shape of the bony labyrinth, but they contain, again, inside them, they contain fluid, which is called endolymph. So there's fluid outside, which is inside the bone, which is called perilymph, and then a fluid inside the membranous labyrinth, which is called the endolymph. And we have the semicircular duct, and distended by an ampulla. Probably we have mentioned this word ampulla before. It's a distension of something. We call it ampulla. So distended by an ampulla, semicircular duct distended by ampulla. And then the vestibule has two portions. It has a region which is called utricle and a saccule. So the utricle and saccules are part of the membranous labyrinth, which are present in one bony cavity, which is called the vestibule. So the bony cavity is the vestibule. Inside it, just imagine that there are two balloons containing fluid called utricle and saccule. And inside these balloons, inside this fluid, there are the hair cells, the sensory cells. And then within the cochlea, we have the cochlear duct. Again, it is formed of membrane. It is part of the membranous labyrinth and contains endolymph. Now let's take a section in the cochlear duct. If this is the cochlear duct here, I'm going to have a section in one of the turns here, in one of the turns of the cochlear duct. So as I have just said, that this is the bone, okay, and inside it there is the membranous labyrinth. Here we have fluid, which is called perilymph, and this is here again there is fluid which is called the endolymph. Okay, the modification here is that this membranous labyrinth is not circular in section, but it is more like a triangle. So that's why we have perilymph here, and then the endolymph was located in the cochlear duct. It is not exactly circular in profile or in section. It is not circular in section. This is what we call the cochlear duct. Now, if you follow this three-dimensional view, you will find that at the tip of the cochlea, these two parts, they are going to communicate with each other at the end, because at the end of the bone, that will be the tip of the cochlea, and that will be the tip of the cochlear duct. Okay, so that's why this scala vestibuli will communicate with the scala tympani at this location. So they are communicating with each other. The fluid which is present here is the same as the fluid that is present here. Any ripples that are produced here are produced there. Any waves that are produced, okay? They are communicating with each other. So in section, the cochlea has two scalas, a scala vestibuli and a scala tympani. The scala tympani is continuous with the round window, the window that is not closed by bone, but is closed by membrane. And that's why if there are excessive vibrations of the fluid in the scala tympani or in the scala vestibuli, they will be taken up, they will be equalized by the movement of the secondary tympanic membrane. These vibrations are produced by the movement of the foot plate of the stapes, which is present in the oval window. In other words, the oval window communicates with the scala vestibuli, and the round window communicates with the scala tympani, and then both these scalas, they are communicating with each other at the tip of the cochlea. And in the middle is the cochlear duct. So the cochlear duct is separated from the scala vestibuli by a membrane, which is called vestibular membrane, and is separated by the scala tympani by the basilar membrane. Now let's go a little bit deep. You will find that these are the hair cells, the cells that are sensitive for change the mechanical waves into nerve impulses. So they have hairs. These hairs are either stereocilia or acilium. Stereocilia means long microvilli. And there are supporting cells as well. Both of them, they are present on the basilar membrane. And this is what we call the organ of Corti. The name of this is called the organ of Corti. And it is covered here by a membrane, which is made of gelatinous material called the tectorial membrane. So if there are any ripples or vibrations taking place in the scala vestibuli, these will be converted into vibrations in the basilar membrane. So basilar membrane is going to vibrate, and this is going to cause the hair cells to move. And as you can see, the tips of the hair cells are embedded in the gel. So therefore, this will cause bending of the hairs of the hair cells, and then signals, electrical signals, will be transmitted to the nerve endings of the first order neurons of the cochlear nerve. I have just mentioned that. The first order neurons are located here within the bone. Again, everything is covered by bone. So you have bipolar neurons. They have their central processes here, and then their peripheral processes will produce, or axons will produce, the cochlear part of the vestibulocochlear nerve, the 8th cranial nerve. Let me show that to you here again. So we have the auricle of the external ear, which collects the sound waves, transmits them through the external auditory meatus, and this is going to cause vibrations in the tympanic membrane, and then vibrations in the malleus, incus, and stapes. Excessive vibrations are going to be controlled by muscles, stapedius and tensor tympani. Okay? Now these vibrations here, which reach this stapes, the stapes closes a window. Deep to the window, there is fluid, which is perilymph. So vibrations of the stapes will cause vibrations of the fluid, and the vibrations of the fluid will be transmitted from the scala vestibuli to the scala tympani, because, as I said, they communicate at the tip of the cochlea. Excessive vibrations of loud sounds are going to be absorbed here at the round window, because the round window is closed by a thin membrane, a secondary tympanic membrane. While this fluid here in the perilymph is moving, it is going to cause movement of the basilar membrane, and will cause movement of the hair cells, whose hairs are embedded in the tectorial membrane. So the hairs are going to bend, and this will start the electrical signals. Okay? The electrical signals are taken by these neurons, and which are going to form the cochlear part of the vestibulocochlear nerve. So here we have sounds that have higher frequency. They are going to stimulate the organs of Corti that are present in the lower turns of the cochlea. While low-pitched sounds, they are going to stimulate the organs of Corti that are present in the upper part of the cochlea. Lower part of the cochlea, we have the membranes. They are a little bit tight. They are not that flexible. So they cannot be stimulated by low-pitched sounds, but the low-pitched sounds vibrate the more flexible upper part of the cochlea. And the loudness of the sound will be translated as the number of hair cells that are going to be stimulated. The more the hair cells in the organ of Corti that are, so for example, if it is a sound is not that loud, then probably it might stimulate one of these hair cells. But if the sound is so loud, then it's going to stimulate all the hair cells, and this will be translated by the brain, or perceived by the brain, as loud sound. And this is how the auditory pathway takes place. So from these cells, which are present in the spiral ganglion, we call it, these cells in spiral ganglion, their axons will go to the medulla oblongata. This is the region of the medulla oblongata here. And there are cochlear nuclei in the medulla. And from there, we have second order neurons are going to project. Mainly they project to the thalamus, but some of them they project to the inferior colliculus of the midbrain. You remember the inferior colliculus of the midbrain? What is the function of the inferior colliculus? There is a superior colliculus and inferior colliculus. it is concerned with the startle reflex. Oh, remember the startle reflex? So some of them will end here in the inferior colliculi to serve for this reflex, and then others, they are going to, definitely they should go to the thalamus, where it is the hub of all sensory information. And then from the thalamus, third order neurons are going to project to the cerebral cortex. Which part of the cerebral cortex that serves hearing? Auditory part. Where is it located? In which lobe of the brain? In the temporal lobe of the brain. It is located in the temporal lobe of the brain, the upper part of the temporal lobe of the brain. Some of the fibers cross, some of them do not cross. Therefore, each cerebral cortex receives fibers from both ears, not necessarily from the contralateral ear. Now, when I'm saying first, second, third order neurons and the cortex and lobes, I'm assuming that you have studied that from the previous lectures. The last part of this lecture will be about the vestibular apparatus. And again, like in the hearing, it consists of a bony labyrinth and a membranous labyrinth. Probably I gave an introduction to this before. So we said that this is the bony labyrinth, semicircular canals, and contain the semicircular ducts and the ampulla. And inside the ampulla, we have the sensory cells for equilibrium. And then we have utricle and saccule. This is the saccule, utricle and saccule. They are present within the vestibule, which is the bony parts. Again, the utricle and saccule, they contain sensory receptors for equilibrium that are a little bit different from the sensory receptors which are present in the ampulla, and we will see that in a moment. So we have two types of equilibrium or balance. We have static balance, static equilibrium, and dynamic equilibrium. The static equilibrium maintains the body position, maintains the head. So when I tilt my head on one side or tilt my head forwards or backwards, there are certain receptors which are located here in the utricle and saccule that are going to be sensitive. But we have another equilibrium, which is the dynamic equilibrium, which is equilibrium during motion, during rotation. And this equilibrium during motion is detected by receptors which are present in the ampulla of the semicircular ducts. You can see them here. This is for the dynamic equilibrium, and this is for the static equilibrium. We have receptors. They are not exactly the same, so we are going to study their structure here. So let's start with the utricle and saccule. The utricle and saccule are located in the vestibule. The vestibule is the bony compartment, and it contains utricle and saccule. Again, this bony compartment has perilymph. Utricle and saccule, they have endolymph, but in addition to that, they have thickened regions, which you can see them here, called the macula, which have supporting cells and have hair cells. These hair cells, they have a cilium and they have stereocilia. The term stereocilia means long microvilli. Unfortunately, they didn't change it, but because at the beginning they thought that they are cilia, and then they discovered that they are not cilia, so they call them stereocilia. Okay, so they are microvilli, and we have the cilium. Let's say these are the hair cells, and these hairs are embedded in a membrane. The membrane is produced by the supporting cells. The membrane is called the otolithic membrane, and on the surface of the membrane there are small tiny crystals called otolith. Oto means related to the ear, oto, O-T-O, not A-U-T-O. O-T-O means related to ear, and the lith means stone. So they are small tiny stones that are present in the ear, in the inner ear, but not related to hearing. They are related to equilibrium. So you can just imagine that if you tilt your head, then what's going to happen to the otolith? They are going to move with the gravity, and they will cause vibrations in the otolithic membrane, and this vibration in the otolithic membrane is going to cause bending of the hairs, and therefore signals, electrical signals, which are going to be transmitted by the nerve endings connected to the hair cells. Okay? So these structures, they are present in the utricle and the saccule, both of them, in the utricle and the saccule. And they are perpendicular to each other. So that's why they can detect, for example, side movement and forward, backward movement. So utricle and saccule are perpendicular to each other. You can see how this works. When the head is tilted, then the force of gravity will draw the otolith with them, bending the membrane, otolithic membrane, bending the hairs and creating the signals. The difference here in the semicircular ducts and their distension, which is called the ampulla, we have an area in the ampulla which is again thickened, and it's called the crista, not the macula. Here it is called the macula. See, this is the macula, which is here it is called the crista. And again, there are hair cells with a stereocilia, with a cilium, and they are covered by a gelatinous material, but there are no otoliths. So the difference here is that there is no otolith. What do you think that is present in this area on either side of this gelatinous material, which is called the cupula, like dome? What is there? Is it air? Is it fluid? Is it gel? What is inside the membranous labyrinth? It is fluid, endolymph. And this endolymph has a consistency that is different from the consistency of the gelatinous material. Okay? So during movement, during rotational movement, as you can see it here, during rotational movement, the hair cells are going to move with the bone, but the gelatinous material is going to lag behind, and therefore they are going to cause bending again of the hair cells. And you can see that we have 5 ampullae, so this can detect the movement in 360 degrees in one way or another. It's a very complicated system. And this is the basis actually of what we call the motion sickness, when the brain receives conflicting signals. Like for example, you are in a boat, the boat is moving, the brain is receiving signals from the ampulla that there is movement, but when you are inside the cabin and you are looking outside, you don't see that there is any movement. It's all the same. Okay? So the brain will receive conflicting sensations that there is no movement, and this will result in dizziness, nausea, vomiting, and things like that. Yes. So again, here these impulses from the hair cells of whether the crista or the macula, they are going to be transmitted through the vestibular branch of the vestibulocochlear nerve, and they will go to the brainstem, partly in the pons, partly in the medulla oblongata, and then they are going to project into, some of them they project to the thalamus, and from the thalamus they project to the vestibular area in the cerebral cortex, which is most likely it is in the insula. If you remember the insula, that's hidden part of the cerebral cortex. However, some of the projections will go to the cerebellum and the spinal cord.